Jonah, who's gone, can watch this, and all the other people who maybe need to do some last minute cramming, you can listen to this as you walk to the final, if you wish. 2 times speed, perhaps. I'll listen to it while I'm in lab, maybe, except probably not. Huh? Is it? 7 p.m. Oh, I don't know. I have not checked. The people say it's in Corey. It's 7 p.m. to 10. Who knows if we'll need the whole time, but he did suggest it might be quite long.
I thought he said like it was like an hour and a half.
Yeah,
Okay.
he said it would take around an hour probably. But I always use the last minute, so.
Yeah. Okay, anyway. Introduction. You should everyone, I think he explicitly said this will be on the final.
Yes.
Algae are generally oxygenic phototrophs that are not embryophytes,
Not plants.
which are land plants. So you gotta have your general, you gotta have your oxygenic, you gotta have embryophytes. Not that. Or not land plants. Both are acceptable. They evolved like this. Yeah, this is not that important, I feel. But I guess it is important to know that they are polyphyletic. It does not include the common ancestor, which kind of makes sense because it includes both cyanobacteria and like eukaryotic algae, which includes the eukaryote that engulfed the cyanobacterium, and that guy was not an algae before it did that. So how could it be monophyletic or paraphyletic? It can't. Boom. Okay, you should know those terms. I'm not going to explain them because you should know them. Yes, yes. Also, please ask any questions as they come up if you wish. We talked a little bit about genetic and genomic tools, forward and reverse genetics. You go forward from the phenotype to genes, and reverse is going, so like you have a candidate gene. This is more of a modern technique as we got good at biotech and we're like, oh, I alpha folded this protein and I think it might have to do with this. Let's knock it out and see what happens. That's reverse genetics. Chris is a big believer in the old-fashioned forward genetics, as he has shared and is very prominent in his research. But in the lab section, that was kind of reverse genetics. We were like, here are these genes that we know of, and let's see what happens. So that was a thing that happened. Not going to be on the exam for sure, but like if you want to think about it. Anyway, we talked a little bit about this experiment. I don't think this is going to be on the exam. It was clammy paper, but it's kind of just like understanding experimental design. Maybe you should know Western blotting, but I don't think you should like really worry about it. But like, okay, the idea is like, is the protein there? And we can find that out by like isolating the proteins. So you get all the proteins, and then you can do a gel of the protein, so spread them out, and then you apply an antibody that is designed for your protein. Sorry, this picture is not the highest quality. But since it's designed for your protein, it'll bind to your protein, and then you can have a secondary antibody that binds to the first one, because designing a fluorescent antibody is expensive, and if you have just like a general one, it can bind, you can have like your first antibody, the part that you designed special for your protein, and then another part that's special for the secondary antibody, and this is going to be like a very general one. This saves you money instead of designing a secondary antibody that's fluorescent that also binds to your protein. So this one is just this extra step makes it a little bit cheaper. And then you can see hello means this one is fluorescent or something, some sort of visual representation that shows you where your protein is. So is it there? Yes, if it glows, or fluoresces to be proper. And then here are some more terms. We have homoplasy, which is basically just convergent evolution, because they really had to make up another term. Parsimony is used when we're talking about kind of like genomic data. It's used in phylogenetics. It's like how similar they are. So ideally conserved DNA. So like if you're looking at homology between a protein of different species, something that is more parsimonious, more homologous, more similar, more closely related. So when you're making a phylogenetic tree, you want the most amount of parsimony. So your most similar things are most close together, because that makes sense. Also, you can get more information if you concatenate your proteins, because maybe one protein, you can see the conservation there, but one protein is not that much data. So if you have a bunch of conserved proteins and you put them all together, you glue them together in your data set, you can get more information. We'll talk about that a teeny bit more when we talk about the papers. We talked about cyanobacteria. The only thing that I feel like you really need to get to know, or like take away from this section, we'll talk more about it in like nitrogen fixation, cyanobacteria, I mean, but the only thing that I would say is really important is filamentation is an example of homoplasy, convergent evolution. It is not a monophyletic trait, whereas heterocysts, which is nitrogen fixation, is monophyletic, evolved once and has been kept in its people. That's pretty cool. Okay, then we talked about phylogeny, taxonomy. I think that honestly, I really liked Chris's, let's see if I can find it. This is not a very, hey.
It's a 60-page review slide.
Yeah. But he kind of consolidated it in a way that I found to be helpful. Because I don't think, he did say on his review, like you should know the names of the different 11 phyla. Where is
So that would be given a list, you don't have to like...
Yeah, you should know be able to actively recall the names, but you should be familiar. So I'll show two graphics that I thought were helpful. Where are they? Did I pass it? Aha, thank you. It's this thing. So we start with cyanobacterium. It is engulfed by a eukaryote. We'll go over that endosymbiosis a little bit more later. And then we have our first eukaryotic alga, and then this is our primary endosymbiosis. This diverges in 3 ways: chloroplastida, rhodophyta, and glaucocystophyta. And then let's look at that other, sorry, I was not entirely prepared for this, but I ain't getting paid, so you're just gonna have to deal with it. I liked our way of demonstrating this here. Okay. Once again, high quality image. From cyanobacteria, we have our primary endosymbiosis here, and these 3 Archaeoplastida phyla. Chloroplastida, this is the one that turns into our land plants eventually. Which group turned into land plants? Is that just like a separate, we don't talk about it because it's not algae? Okay. Imagine another arrow is not algae. It has chlorophyll B, so all of the things that either came from chlorophyta or engulfed a chlorophyte has chlorophyll B, or I guess could have. I don't think that they've lost, I don't know, that's not within the scope.
They all have chlorophyll.
Okay, they all have chlorophyll B, and they've lost the, is this supposed to be PBS? I'm guessing this is peptidoglycan. Oh yeah, this, because I always refer to peptidoglycan as just PG, but peptidoglycan, I guess let's talk a little bit about endosymbiosis right now, because I think it just is a good time to talk about it. Chris's thing was good. So the cyanobacterium that was engulfed by our first eukaryotic, not algal, but becomes algal ancestor, was this cyanobacterium was gram-negative, meaning it had 2 membranes, which is why we think of our chloroplast as having those 2 membranes, although I guess there's also, 2 membranes, and then there's also the outer thing that gets eventually away. That goes, like, the vacuole, that is, we don't think about her, she gets evolved away. So in those 2 membranes, normally bacteria have peptidoglycan as a cell wall, but that has been evolved away. This is great. In the chlorophytes and the rhodophytes, but not the glaucocystophytes. So that's interesting. There's also nobody that we have, or that we know of, I guess, that evolved from glaucocystophyta. It's just chlorophytes and rhodophytes. Oh, I scrolled away from the thing. But Jonah's version of this was pretty good too. But I liked Chris's because it's just like all here. And he also emphasizes the presence of a nucleomorph in chloroarachneophyta and cryptophyta, which you can remember that they both have, they start with the letter C, if that's how you want to remember it.
Or you could be like, ooh, nucleomorph, that's kind of creepy.
Kind of creepy. Spiders.
And spiders.
Yeah, it's creepy. I like that. So a nucleomorph, yeah, sounds kind of a scary word, but it's just like leftover nucleus from your endosymbiont. Yeah, and it's a nice little token of evidence of endosymbiotic theory. So if that's a question, it's like, what's evidence of this? Nucleomorph in my creepy cryptophyta, chloroarachneophyta. And it also happens on two different, like from two different clades. So that's like, look at that happening.
Yeah.
Oh, and also we don't talk about her.
Example of the homoplasy.
Homoplasy. We don't talk about her, but there's a second primary endosymbiotic thing that we don't talk about in this class at all, which is polynella. Pretty cool though. It's happening again. Mu and endosymbiosis. So yeah, so our chloroplastida, the only things that we really talk about are euglenophytes and chloroarachnophytes. I don't know why I have such a hard time saying that. And then rhodophyta has like everybody else, so ochrophytes. So these are our photosynthetic stromatophiles, our diatoms. Haptophyta, cryptophyta, dinophyta, apicomplexa. Apicomplexa is that group with like 2 photosynthetic guys, including Plasmodium, malaria.
Especially like malaria.
Yeah. Dinophyta, we've got our Symbiodinaceae, coral relationships. Cryptophyta has a nucleomorph. Haptophytes, they got stuff too. I don't remember. Moving on. Hopefully that's satisfactory. I don't know if it truly is, but here, let's look at some more things, I guess. I don't know if I really want to. Yeah, the only thing we have about chloroarachnophyta is nucleomorph. I definitely don't think we need to know how many species there are. Although it is interesting that there's only 2 photosynthetic ones in apicomplexa. Yeah. Oh yeah, rhodophytes lost their flagella. I have so many things. Rhodophytes lost their flagella. It's the only group that fully in all of the things have lost their flagella. And you might be like, chlorophytes, like land plants, they don't have flagella, but like some of them do, like clammy.
Haptophytes are chalk.
Oh. Haptophytes are chalk.
Haptophytes are chalk.
And the chlorophytes also lost their phycobilazome. They're the only ones who have done that, really? This is kind of surprising.
I mean, some of the like
fully, I mean.
Some of the like later like secondary endosymbiosis,
Yeah.
so red algae like don't have a phycobilin, I think. I don't know which ones, but
Well, cool. Moving on. So that's our taxonomy section. Yeah, chalk. Second most prolific algae, followed by, or secondary to who? Who's our most prolific algae?
I think it's a haptophyte that has the mucin A in it.
That's very
I've talked
possible.
about this before.
But who's number one prolific algae? We should know this. Diatoms.
Is it really diatoms?
It's definitely diatoms.
Oh my goodness.
Yes, most prolific. You got it right there. Bless you. Okay, so more on symbiosis in the IGI whiteboard. Yeah, so I feel like this is like kind of obvious. Like, what are the common themes? Oh, they share stuff. They talk to each other. They gotta exist. You know, climate change happens. That can affect them. But when we're talking about like types of symbiosis that we talk about in this class, not endosymbiosis, which for some reason I feel like this should have been before, but we already kind of went over this. We talk about some examples of like algae working with other species, so not like a full endosymbiosis, just a symbiosis. So you can be working alongside, or you can be working in. Working in is just corals. So the coral gives a home to a dinoflagellate, which donates 95% of their photosynthetic products, which I feel is very generous. And they give, the photosynthetic products, and they get protection from light, and they get some inorganic nutrients in return. So thank you, corals. Fun fact, completely irrelevant. My friend's mom got, was scuba diving or something, and then scraped her, I guess this was scuba diving, I don't know, she was in a beach, and she scraped her foot on a coral, and she was like, it's fine, and then
it's never fine.
it grew in her. So she had to have surgery.
Yeah, so every time I hear about somebody scraping something on coral, it's like fucked up.
Yeah.
Something bad happens. Go to a doctor if you scrape coral, or if a cat bites you really deep, their teeth are really deep. It can be bad. You can swallow it.
Not recommended. Anyway, but our extracellular relationships, we talk about the Azolla-Anabina relationship, where Anabina, which is a cyanobacterium with heterocysts, it's going to give nitrogen. Azolla is a plant. It can actually give the carbon, which is unlike the other relationships we hear about with algae. And so it just sits in little cavities on the leaf of the fern, and it inhibits this enzyme glutamine synthetase of Anabina, which tells it like, oh, we have enough fixed nitrogen, and it says since it's broken, it's like, well, we need to keep making more.
Yeah, that's how it eats the nitrogen, and so.
Yeah, it uses up the nitrogen, but since it can't use it, it'll make more forever.
Yeah, it's in the permanent I need more nitrogen mode, and so it can only get nitrogen from the azolla.
Yeah, because the thing that it would make signals that we have enough of it, but it can't make the thing.
Would it be more of a commensalism or a parasitism?
I mean, theoretically, this exact interaction is parasitic, but this full relationship is helpful for the cyanobacterium. That's why it happens. It evolves this way. So like, I don't think it's necessarily like bad for the cyanobacterium. It's just using the cellular mechanisms that would normally regulate it to not. You know.
It's like.
It gives stuff in return.
If you think about friendship too hard, it's like, oh, I do this thing because I want this person to be nice to me. It's manipulation.
Sure, yeah.
This
But they
is
still
the
pay
equivalent.
something.
Yeah, exactly.
Yeah. And being is passed on to new generations, so that's pretty fun. So if it were evolutionarily not advantageous, this probably wouldn't happen. So clearly there's something good going on here. Lichens are a fungus and an algae, and there's always, or I think Chris said most of the time, but I heard from another professor that there's always a basidiomycete yeast, and we don't really know what it does, and it's definitely not going to be a question, but I think it's cool. And it's another, I give you sugars as my algae, who's a phototroph, and then I get protection from both light and desiccation, so I won't dry out, which is why lichens can live like in the desert, can live in crazy places. We saw it all over the beach when we went to the beach.
Did we see lichen at the beach?
I remember seeing lichen at the beach.
I didn't notice too much of it
Well, down on the beach part of the beach, I think it was like
Yeah.
not lichen.
Yeah, but we saw those trees that was like hanging with them.
Up in the trees, 100%
Yeah.
lichen.
Yeah.
Fun fact, California state lichen is
Yeah.
like, I don't know if other states have a state lichen, but we do.
Mhm.
They don't. We're special. That's
Wow. Wow. We are going to go over the Porphyra life cycle at the end fully. Like, I'm not going to just, like, we're going to kind of talk about it now, but, like, we'll give a full, and that probably won't be in this recording
I was
because...
doing it yesterday and I was so confused.
Oh, no. Okay. We'll go over it. We'll go over it. This is not really the Porphyra life cycle. When we were doing this study session for the quiz, he had implied that it wouldn't be on the quiz, but rather the final. Also, all of these pictures are available on the Discord somewhere, as we sent them, you know. So if you want any of them, or you can, whatever. Anyway, life cycles. Okay, the way that you should think of these, I feel like the way that this was explained was not entirely intuitive. Haplontic means I do mitosis in my haploid state. So I grow, I develop as a haploid alga. Diplontic is like us, we're humans. We do mitosis in our diploid state. You know how we are all diploid right now? And we don't divide as like a gamete. Like the sperm and egg just stay sperm and egg until fertilization. We are diplontic. There are some guys, though, that are diplohaplontic, which divide in both. Is clammy haplontic? Or it might be diplohaplontic.
Like
Clammy.
I think it's different.
Well, it's not. I don't know. But this is the land plant life cycle. This is the us life cycle.
Planting is haplon.
This is the climbing life cycle. There we go. And it does meiosis when it is not in the one that it isn't named after. So
Zygote is its only diploid form.
yeah, zygote is only, yeah, okay, makes sense. Thank you, team. Oh yeah, and there are some terms, isomorphic and heteromorphic, which are named aptly. However, there are other terms that are also similar, so it's kind of confusing. But if you really look at the word, it kind of makes sense. Like isomorphic. So iso meaning same, and morphic meaning like morphology. They look the same in their diploid and haploid forms, and heteromorphic is they do not look the same. But then you talk about isogamous and anisogamous, which is the same principle but applying to the gametes, which is indicated in the word gamous. So the male and female look the same or different. I don't know if that ever made it on this board, but there you go. Oh, I think it might be somewhere. Aha, maybe. I don't know. Anyway, so yeah, these are the three that you should know, and this is the one that Porphyra is, and we'll go over that again later. Algal life cycles. Oh, diatoms. We'll talk about this more later. Porphyra. Oh, we did draw this. Yeah, we'll do this later. Yeah, monoecious means that one gametophyte makes both the male and female gametes, and dioecious means that one gametophyte makes the female and one gametophyte makes the male. Porphyra is monoecious. Fun fact. Oh, you should know, like, fucus, how the zygote develops. There are, like, from the zygote you get asymmetric division into the thallus and rhizoid. The thallus, which becomes like the leafy thing that is the alga of the alga, and the rhizoid is kind of like the root-like structure that is still of the alga, but like it's not the part that we think of when we think of algae, our macroalgae, our seaweed. And so the way that that's oriented is obviously you want your roots to grow down, to grip into the soil or the rock or the whatever, rather than like roots growing into the abyss. That seems a little silly. So the way that it figures out where down is they sense the light, and calcium channels will go away from the light, and that will orient the rhizoid away from light. Boom, into ground. Fantastic. And then we talk about volvox. And there are two different types of cells. There's the ganidia, which are the primary like photosynthetic guys. They're harvesting lots of light energy, and the somatic cells, which do some photosynthesis, but mostly their job is to make the extracellular matrix, which holds them all together. Volvox is like a later evolved version of clammy, kind of. So each little guy in there looks like clammy, which is kind of fun. And we'll talk about multicellularity in a little bit. I literally don't know what to tell you about this. I'm very confused by it.
I just don't know why it was mentioned.
I don't understand it at all. I don't know the significance of it. I don't know what it means. I don't know all of it. So this is like, yeah, no. Moving on. Symbiosis again. I guess we reviewed this again. Diatoms. Oh, you can tell that Jonah wrote this. Look at this beautiful, yes, hella diverse. Silica. Okay, so yeah, now we're talking about diatoms for real. Let's talk about how they divide. So diatoms are basically a petri dish, but they have, like, you could say parafilm in between, which are the girdle bands. The top one is the epivalve, and the bottom one is the hypovalve. You probably don't need to know those, but you should know valves are the top and bottom, and they fit into each other like a petri dish. And when they divide, it's like one valve. Well, both valves are freed, and then the other valve is formed, but the other valve will always be the smaller of the two, as kind of demonstrated here, with the purple being the new valve. And so at some point, they become too small. There's a threshold reached around 30% of the original, or 30% reduction, I think, of the original size. Or maybe it's 30% of.
They get small.
They get smaller at some point, and then and then they're like, let's start over. And so I don't know how they do it, but they start over. And they get big again. Via sexual reproduction, thank you. But I don't know, like, mechanistically how that works. But yes, they do sexual reproduction and then they start over. And they do lots of photosynthesis, very important. Yes. So how do they make the silica? There are like three main things that we talked about, which are, it's amorphous silica, which forms in silica deposition vesicles. And when I was reviewing Chris's notes, can I just quickly, I don't know, SDD, yes. He said there was two types, the valves and girdle band, so I don't know if he's saying that the valve itself is a silica deposition vesicle, but I guess that kind of makes sense, because like that's where you'd want the silica. Oh, okay. Okay, there you go. And then some other things that we talked about is silicanin, which is a protein that's essential for frustral biogenesis somehow. This is a paper that we read also. It's the one where they poked it and it got squishy when they got rid of silicanin.
Do they talk about silicanin or silafin?
So silafins are a different thing. They help precipitate silica. I think that's better understood. And also it has to do with low pH.
That was the best paper.
It was so good. I loved the poke.
I love the poke. That really made the paper. My favorite paper was the Jonicus paper, but I think I'm a little biased because I just really think the Jonicus lab is cool. But yeah, it got squishy when they got rid of the silicanin. Yeah, the actual function of silicanin was not really discussed, but that's okay because you see here that he just wrote that it's involved.
It's involved.
So, what he might ask, what might you expect if you knock out silicanin? And you could say it gets squishy. It gets squishy. But not completely squishy. It's not like a little squeaky. It's yeah. But if you poke it, it might not bounce back all the way. We talked about this already. Porphyra life cycle. We'll get to it. We talked about this. We talked about that. Yeah. Yeah. Multicellularity, is this where we, wow. How many times? Oh. No, this is a different, okay, cool. Okay, cool. I guess we don't really talk about, you can see when we make the transition to studying in Evans. So yeah, multicellularity has evolved multiple times.
I think this one was in the bottom of Evans, wasn't it?
No, this one was definitely in the IGI.
One of them was in the bottom of Evans.
That did happen once, but I think that was 160.
Was it 160?
I believe so. Strongly.
I don't remember.
Anyway, yes. Multicellularity, responsible, like there's just a few genes, honestly. They did that paper that we talked about in class, but we didn't read the paper where they put it with was it clammy? Or I think it was a relative of clammy, closer to Volox.
I think it was clammy.
I think it started with a G. I remember G very distinctly, and I think in my notes there's a hole where the actual name of it was supposed to be, because I couldn't write it down in time. But anyway, they put it with a predator. I also don't know the name of it, but I do know it starts with a P. And they just did that over and over again until it evolved multicellularity, because when you're big, you're harder to chew. That's pretty cool. So,
The RBG.
yeah,
Different one.
riboblastom, retinoblastoma. Sounds right. Okay. Motility. How do we move? So we talked about like flagella. In most of these examples, we have this 9+2 conformation where there's 9 outer doublets on the outside of the flagella and a central pair, which is like 9 doublets and then 2 distinct ones. So you can kind of think of it as 18+2, but I don't know if that makes it more confusing or less. And these circles are microtubules. And most of the guys have this conformation except for diatoms. So diatoms don't typically have a flagellum, but their male gametes do, because they got to be, moving around. They are
9 and 0.
Yeah, it's 9 and 0. They don't have the central pair in the middle. What is the term for when it moves around or doesn't move around?
Motile?
Well, yes.
Non-motile?
No, it's specifically referring to. Yeah, it's something over here, but I don't think we wrote it down.
I'll do some poking around.
Okay.
Find it.
If it's found, we'll report back.
Okay.
But centric diatoms, they still want to be moving, and they move kind of like, I feel like kind of like a starfish. They have those little. They look like they're just sitting there, but underneath they have all these tiny little legs, but they're not actually legs. They're raphy in this case. I don't think that the starfish little suckers are called raphy. Yeah, only for the pennate. The centric ones, I guess, just sit there. But the centric diatoms do have this flagellum on the male gamete, but else they do not. But the pennate, they move like back and forth like this. They don't like move sideways, which I think is interesting. I don't think there's gonna be a question about that. And so yeah, it's like you can imagine. This is the pennate diatom, and they got these little extensions bound in the plasma membrane and the actin, and these little raphy just squirming about. Okay, cool.
So for the algal life cycles, if the gametes are isogamous, they look the same.
Yeah.
And isogamous, they look different.
Yeah.
And if they're oogamous, then they have the typical non-motile egg.
Oh.
There's
Okay.
also
So
like,
I
the
guess that's what that's.
between the zoospores for asexual reproduction, but that's kind of that's
I think I was looking for oogamous, which I do think was on that thing that I was looking at. That's what these centric diatoms are, but I don't know about the pennate ones because we don't talk about the male gametes having flagella, but we do know that these do, so I'm guessing they're not. But I don't know. Anyway, biogeochemistry. Wow. This was not my favorite one. There was a great oxidation event around 2.4 billion years ago. Me when I can photosynthesize cyanobacteria, #cyanobacteria. And you better know this one. You better know that algae are responsible for 50% of global net primary production, because he will ask us about it. So what is net primary production? It is the net, so the gross minus what is lost through respiration. So GPP, gross primary productivity, minus like cellular respiration. So 0.2% of total photosynthetic biomass is stored like in the fossil fuel thing, but about like 10 to 30% sinks through the biological carbon pump. So why is this number so low? High turnover rate, algae be getting eaten all the time, and they die really fast. But some of it does sink, and there are different things that, sorry, when I said the 0.2, I meant the 0.3% makes it the oil. This is referring to like how algae are super, super light, but they are doing so much of the photosynthesis. So that's kind of crazy. Very efficient little guys. And there's a bunch of ways you can measure this. You should probably memorize 1 or 2 just so you have something to say if this was on a quiz. I like satellites measuring chlorophyll concentration. I think it would be really silly if you said sensors detect the depth of the euphotic zone, because what even is that? Like, we know what it is, but like, come on. I don't know. I just feel like that's a silly one. Climate changes. Boom. How do algae store stuff? They have carbon polysaccharides. I don't think this is that important of a question. We should know some cool algae products, which we're going to talk about at the end, which are like our alginates, our caragenins, our agar. That's all of them. But we'll talk about that slightly more at the end. Carbon fixation. You definitely don't need to be that familiar with the carbon cycle. This was kind of a review because Jonah was like, whoa, and I was like, how do you not know Rubisco? But Rubisco, you should definitely know Rubisco. It stands for RUBIP, ribulose bisphosphate, carboxylase oxygenase, right? Meaning it can fix RUBIP with both carbon dioxide or water. Nope, not water, oxygen. I don't know why I said that. And that's a problem because if you fix it with oxygen, you're gonna make p-glycolate, and that's stinky, it's toxic. Why would you even do that? It requires energy to fix instead of giving you energy. You're wasting your precious RUBIP, which you have to go around the cycle an extra 2 times to get one single half of a sugar, or one sugar I think, or maybe, no, 2 times to get 1/2 of a glucose in your G3P. And that's silly. Why would you do that when you could just fix it with carbon dioxide? And RUBIP, all things considered, or Rubisco I mean, is all things considered pretty good at that. There's way more oxygen out there than there is carbon dioxide. It's like 30% of our atmosphere, right? Question mark? Oxygen, but CO2 is way less. So it's pretty good at it, but it's still not that good at it. And so what the solution is for our algae, who are very efficient, some algae that is, so there are cyanobacteria which have carboxysomes where they kind of shove bicarbonate in, which is just like easier for Rubisco, and that it won't diffuse out of the carboxysome, so it concentrates it in there, so there's a higher concentration of it, so Rubisco can convert it easier because there's an enzyme that takes that bicarbonate, carbonic anhydrase, and turns it into carbon dioxide that once it's in the carboxysome, it won't escape, and so the Rubisco can do its job easier. And then our eukaryotic algae have our pyrenoid, which we read a paper about. That's the Jonicus paper I was talking about. Which we will talk about later, and yeah, it's like basically the same principle, evolved completely independently though, so that's kind of cool. But I do think that they are like, that's the only times that they've happened, but pretty cool. Clammy has one of a paranoid. Fun fact. And so you should also understand this graph, which we might talk a little bit more later about, but I'll just talk about it like fully now. So. Hello? Okay, so when you have a thing and you have an enzyme, the enzyme will be better at doing its reaction when there's more of its reactants. Boom.
Substrate.
Yeah, of its substrate. So if there's more oxygen around, like I was saying, it's harder for it to fix carbon dioxide because it's like the oxygen's right there. Like it's touching the oxygen. It's like, why would I react with you? So you can kind of see how efficient an enzyme is by measuring its mu max, which is the maximum, like the amount of, like the highest efficiency of the enzyme, depending on the amount of substrate it has. And so it's this is kind of like a logarithmic graph, so I don't know if you ever really reached this number, so we use the half Umax, and you see how much substrate is necessary to get to that. So if you had, and this was an experiment that we're going to talk about later, if you had another enzyme that has a graph like this, where it has, at the same Umax, it needs a greater concentration of that substrate, so this would be a different value for Km, it would be less efficient. Yes, do we understand that? It needs more of the stuff to do the same amount of work. Thumbs down. Okay, we also talked about light harvesting. At first, Rubisco is at, or sorry, not Rubisco, your photosynthetic rate is a linear graph, but at some point, Rubisco starts getting too busy, it can only work so fast, and so that rate slows down, and at some point, you are, mu and MPQ, you're getting too much light, it's gonna start making reactive oxygen species, you're damaging your algae, it is not good. So your photosynthetic rate will actually go down because it's trying to attenuate all of that and it's getting damaged and it's not ideal, mu and MPQ. A compensation point is when your photosynthesis is equal to your cellular respiration. You should hopefully be doing be above your compensation point. Otherwise you're kind of failing. Engelmann's experiment, this is important, this is gonna be on the exam. How do we figure out photosynthesis? Where is my algae the most efficient? I want to make an action spectrum, so where my photosynthetic rate is greatest by the wavelength of light. And the way they did this was a beautiful experiment. They took this filamentous algae, so what you're looking at here is an algae, a cyanobacterium or something maybe. Oh, actually I don't think it was. I think it was a eukaryotic algae, but I don't remember what. And this is a cell, this is a cell, this is a cell. And so you can see where it is most efficient because these aerotactic bacteria, which need oxygen to survive and proliferate, are greater in this wavelength and this wavelength than like here. So that's cool. And then an absorption spectrum is like a specific pigment where the absorbance is across wavelengths. Yes. So this is the first action spectrum for algae. Very important, very cool. Okay, pigments, different types of pigments. He has emphasized at least twice recently that we should know, we should be able to visually tell the difference between the 3 different pigments. So lock in, guys. Might be something we see tonight. So chlorophyll is a cyclic tetrapyrrole, but it has a phytol tail. Well, not in chlorophyll C. But it's cyclic tetrapyrrole, so it has like a, I mean, this drawing of it is probably not the most ideal, but like, it's very different from a carotenoid, which looks like this. It's a branched hydrocarbon. But a phycobilin is also going to be a tetrapyrrole, but it's going to be a linear one, so it's not going to be this circular shape. So hopefully you should be able to tell them apart. Chlorophyll A is in all oxygenic phototrophs. Chlorophyll B is in our, it's an accessory pigment, and we see it in all of our derivatives of chlorophytes. And chlorophyll C does not have a phytol tail. Don't think that's going to be a question. But since these are both, chlorophyll and phycobilins, are both tetrapyrroles, they share a biosynthetic pathway. That's pretty cool. And so, okay, we talked a little bit real quick. So chlorophyll in land plants, they live in the light harvesting complex, in like this, like how in the antenna to collect light. And phycobilisomes live in the, phycobilins live in the phycobilisomes in cyanobacteria, except some don't have the phycobilisomes, but they still have phycobilins. And what they have is a chlorophyll binding protein, or CBP antenna, instead of a phycobilisome, with their phycobilins, and I guess chlorophyll. So that is interesting. Who has phycobilins? Cyanobacteria, rhodophytes, and glaucocystophytes. Remember, chlorophytes are the only ones that fully evolved it away, as we see here, from our primary endosymbiosis. And the absorbance spectrum complements chlorophyll, so it's very nice when you're living in the water and you have limited light when you're deep in the ocean. So yes. And then carotenoids, these are like our reds and oranges. Also includes puccoxanthins in brown algae. Fun fact. Okay, moving on. Ecology. Phyto, meaning plant. Plankton, meaning what? Floating thing? Ocean thing? I don't remember. That's not a quiz question. there's great diversity. How important do we think that the consumer-producer ratio? I don't know. But this is definitely important. The two major concepts, the competition theory and trophic dynamics. So these are the two major concepts of phytoplankton ecology. So competition theory is like paradox of the plankton. So how do we have so many different types of algae when the ocean is like relatively homogeneous? Like obviously there are different ranges of the ocean. There's different like oceanic biomes, but it's all underwater. And if you think of like the savanna, it's like, okay, there's a tree, you can be living on the tree, you can be living in the soil, you could be living, I don't know, in the air, whereas all the water is kind of touching each other. So it's like, how do we have so many different types of plankton? And the answer that we have decided upon is nutrient availability. And that ties back to that KS mu max, or KM, depending on what you're talking about, thing we were talking about before. And we'll get to that, and there's also a paper on that. But there's also trophic dynamics. So there's two ways that algal populations are controlled. One is top down, so like things eating the existing algae, or like killing it like viruses, or bottom up, which is how the algae can exist, so nutrient availability, for example. And then you should definitely know this graph. You should know this graph. So there's more light at the top. Wow! There's more nutrients at the bottom because it's being eaten by the algae at the top, and it sinks also. And so the deep chlorophyll maximum is where all the algae are. It's that intersection between light and nutrients. And then I don't know how important this is. I'm moving on from this. Do we not, was that our last algae review? Did we not have something in between there? That's the last one I put on the thing, but did we not have one for biofuels or anything?
No, we definitely did somewhere.
You just said that.
Yeah, I think this is the time in which I write on the board. So goodbye to our online folks. We'll come back for papers.